Hello, my name is Alfredo Garcia, and on behalf of Grand Canyon University, I'm going to be teaching you guys about G protein coupled receptor signaling today. So, what is a GPCR? GPCR stands for G protein coupled receptors, and these proteins belong to the largest class of membrane proteins, and they're responsible for most of our senses, amongst other things. So, GPCRs are going to be a type of protein that's found on the surface of cell membranes, so they're embedded in cell membranes, and we'll talk a little bit more about that in a bit. But just a um, quick thing about them is that they really do a lot of things inside our body, so it's kind of hard to pinpoint one specific thing that they do. And if you wanted to try to list the number of things that they do, it would be a very long list. But they are responsible for mediating a lot of our senses. So, for example, our vision and our sense of smell and taste are mediated by GPCRs, and a lot of hormonal responses are also mediated by GPCRs. So, structure. There's currently over 800 known GPCRs in the human genome, and they're all somewhat similar. So they're not all the same, but structurally there are a lot of things that help us identify GPCRs. So they contain 7 helices which span the cell membrane, and they contain their N terminus outside of the cell and their C terminus inside the cell. So quick notation, I guess. N terminus is going to be the start of the protein, and the C terminus is going to be the end of the protein. So all of these proteins are embedded in the cell membrane of their specific cell. The start of the protein is going to be located outside of the cell. The end of the protein is going to be located on the inside of the cell. And another thing that helps us identify them is the fact that they kind of cross the cell membrane 7 times, as you can see here. So they're not structurally this pretty, or as pretty as you see here in this image. Proteins are very long, complicated molecules. If you want to look at the 3D structure of a GPCR, you're going to see that they look somewhat confusing. So they span the cell membrane in a very strange way, but if you do count up the number of times that they go in and out of the cell membrane, it'll always be 7 times. The extracellular portion, or N terminus, of the protein is going to be what binds the ligand, and the inside, the intracellular portion, or C terminus of the protein, is going to be what affects another protein inside of the cell, or the G protein, as we're going to get into right now. So how do they function? GPCRs functions as a means for cells to communicate with one another, and they depend on a ligand for activation. So they're not always active. Something needs to bind to the N terminus of the protein. So the outside portion of the protein has to be bonded to its specific ligand in order to affect an interaction with the inside of the cell. So when they're activated by a ligand, the intracellular tail of the GPCR interacts with an associated G protein. So the C terminus of the protein will kind of change and affect something else inside the cell when you have a ligand bound. So before we move on with G proteins, I just wanted to state just something to keep in mind. GPCRs, or the G protein coupled receptor, is not the same thing as a G protein. So these are 2 different proteins that depend on one another. A G protein depends on a GPCR to be activated in order to affect some kind of change within the cell. So that's a very important distinction to make and remember going forward. Just that GPCR and G proteins are 2 different proteins. They depend on one another and they function together, but they're not the same protein. So they're 2 different proteins. So the G protein is composed of 3 subunits known as the alpha, beta, and gamma subunits. They're sometimes called heterotrimeric proteins because there's 3 different parts, right? So hetero means different, trimeric 3, so 3 different parts to this protein. The alpha subunit of the protein binds GDP, which is guanosine triphosphate, and GTP, or guanosine triphosphate, and they can hydrolyze GDP to GTP. So hydrolyze, if you guys remember, is just a reaction with water which kind of breaks the compound down. So how do they function, G proteins? When a GPCR is activated, so when a ligand binds to the GPCR, the outside part of the GCPR, there's a conformational shape in the alpha subunit of the G protein. So when a ligand binds to a GPCR, it causes the inside of the GPCR to interact with its specific G protein. And what that does is it makes the G protein change conformationally. So the bound GDP, so G proteins normally have guanosine diphosphate bound to them. When they change shape, they're replaced with GTP. So guanosine diphosphate is replaced with guanosine triphosphate. And that causes another conformational change. So there's two conformational changes to the G protein. The first one is caused by the GPCR. The second one is caused when GDP is replaced with GTP. So this allows the G protein to interact with a different protein. And these proteins are usually either ion channel proteins or specific intracellular enzymes, which are just going to be other proteins, right, which function to affect something else. So a quick thing to look at when we look at this picture of a G protein is that you have the cell membrane here, right? So this G protein is also bound to the cell membrane. There are a lot of G proteins, so some of them are going to be a little bit different structurally. Most of them are very similar and look somewhat like this, but there is some slight variation. And another thing to just kind of point out is that this specific G protein is bound to the cell membrane, but not all G proteins are bound to the cell membrane. When these proteins interact with their specific, with the other proteins that they're going to interact with also, I would like to mention, when they interact with ion channels, so ion channel proteins are also going to be membrane proteins, and these usually affect a response very rapidly because they're going to allow some kind of ion or something else into the cell, which can alter the cell very quickly. When you have them interacting with intracellular enzymes, these reactions are going to be a little bit more complicated because they're usually some kind of cascade, and all that means is that the G protein affects an enzyme or another protein, and that other protein doesn't usually directly affect some kind of process within the cell. So it takes a lot of intermediary steps before you actually get to the change in the cell that you're trying to see. So I'm going to go over an example with you guys using epinephrine. So this is kind of complicated if you just look at it, right? But we're going to be going back and forth between this while I try to explain how it is that this functions. So epinephrine, which is sometimes called adrenaline, is a hormone produced in the adrenal glands. So something important to remember is that hormones are messengers that the body produces which travel via the blood. So they're usually produced by a specific cell or organ, and from there they're picked up in the bloodstream, and from there they travel across the body and they target specific cells. So this is already one way in which cells are going to be messaging with different cells. So they're secreted and travel via the blood. This hormone is important in the fight or flight response. So this hormone binds to the B adrenergic receptor located in the hepatic or liver cells and adipose cells. So this B adrenergic receptor is going to be an example of a GPCR. So this is what you have here. Epinephrine binds to the liver cells, and from there you see, so this is the G protein. You can see how it travels through the cell membrane 7 times, and then here on the inside you have the G protein. So this is the G protein coupled receptor, and it's interacting with another protein on the inside, or its specific G protein. You can see that it has its 3 different subunits. So when epinephrine binds here, it causes that conformational change. You get GDP, it's replaced by GDP, which causes a different conformational change, which kind of breaks or separates this protein apart, and part of the protein comes and interacts with another specific protein. So we activate the GPCR activates the G protein, which binds GDP. The G protein separates and activates the target protein adenylyl cyclase. So this is what you see here, adenylate cyclase. The G protein comes and activates this other surface protein, and the G protein separates, or excuse me, adenylyl cyclase catalyzes the conversion of ATP into cyclic AMP. So this is what you see here, ATP catalyzing the conversion of cyclic AMP. So from there, cyclic AMP or CAMP binds to another enzyme, protein kinase A, separating it into active and inactive subunits. So the active subunits phosphorylate the enzyme and to phosphorylase kinase. From there, phosphorylase kinase phosphorylates the enzyme glycogen phosphorylase. From there, glycogen phosphorylase catalyzes the reaction, which breaks down glycogen into glucose. And from there, glucose is transported out of the cell and enters the bloodstream. So this is an important step here, and kind of what I was talking about earlier, or it is what I was talking about earlier, where you see that the G protein, or GPCR, G protein coupled receptor interacts with the G protein. The G protein then interacts with a different protein, and from there you get this cascade. So the binding of the ligand and interacting with the G protein does not cause the specific cellular change that you're trying to see. It needs to activate with a different protein, and from there there's a lot of intermediary steps all involving different proteins, right, till you get to the very last step. So the very last thing that happens in this chemical cascade is going to be that glycogen is broken down into glucose. So this is important in the flight or fight response, because if you're going to fight or flight, you need a lot of energy, and glycogen is stored energy, so we can't readily use glycogen as energy. It needs to be broken down into glucose. So if you bind epinephrine, you speed up this reaction and you produce glucose a lot faster, which means that there's more glucose available for muscle cells or any other cells which may need it in this fight or flight response. Couple of important things to consider. GPCRs deactivate after some time on their own, regardless of having a ligand being bound. So what this means is that epinephrine needs to be bound in order to affect this specific cascade response. But after a certain amount of time of epinephrine being bound, even if epinephrine is still bound, the cascade is going to turn itself off. So it sort of mediates itself. And this is through a separate process. So this process involves a different kind of protein called an arrestin protein. So what this protein does is it sort of blocks off the GPCR. So the GPCR needs to be able to interact with its specific G protein. This other protein, arrestin, kind of comes and binds to the inside of the GPCR and stops it from being able to interact with the specific G protein that it's trying to interact with. So you can still have epinephrine bound here, but if the arrestin protein is bound here on the inside, then it won't be able to interact with the G protein, and it kind of turns all of this off. Another important thing to note, epinephrine has various receptors, which are also GPCRs. So this is something which we've learned about before. Hormones or specific ligands aren't necessarily specific to one specific type of cell. So this cascade, which we were talking about here, is specific for liver cells, which are responsible for breaking down glycogen into glucose when we need it. But epinephrine also binds to muscle cells and a lot of other different cells in order to cause other things, right, which are also important in the fight or flight response. The structure and function of GPCRs may vary So this is something that we've kind of talked about also. So there's over 800 of them, which means that this specific example that I gave you right now is kind of the poster child, or one of the poster child examples that gets used a lot. But if you want to look at different examples of GPCRs, you might find that they function slightly differently depending on the one you're looking at. Another thing is that they're often called a cascade because of the binding of the ligand and the action of the G protein causes a chain reaction of sorts, which, going back to this, is what I showed you guys. It's a complicated process. So the binding of the ligand and interacting with a specific G protein doesn't always cause whatever specific cellular change or physiological change that you're trying to see. It's a cascade. Something sort of important to note here is that if you're trying to affect a GPCR or a process mediated by a GPCR or G protein, depending on the G protein, there is a lot of steps that you can target. So if you're trying to create a drug which functions to affect a GPCR, then your most likely bet, or the easiest thing you could probably do, is either affect the G protein or the G protein coupled receptor. So you can do that via inhibition, so competitive inhibition or other similar means, but you're not necessarily bound to just this specific part of the process, right? There's so many different parts that you can try to stop or interact with to kind of get some different kind of response, right? Because they're usually such a complex response. And that is all I have to share with you guys for the day. So thank you for your time. And if you have any questions, please just comment on my post or email me. Thank you.